All right, welcome back. Let's talk a little bit about enzymes. And the first thing that I want to make sure we're doing is understanding that enzymes are one type of protein. So we know that a protein is one type of macromolecule. So if we just review, we have four classes of macromolecules. We have lipids, we have carbohydrates, we have nucleic acids, and we have proteins. So an enzyme is one of those types of proteins. Now, an enzyme is known as a catalyst, and you may or may not have heard that term before. When I was growing up, before gas grills were really prominent, we would have charcoal barbecue grills that you would put the charcoal briquettes in. And we always had to put lighter fluid, or we put lighter fluid on those briquettes to help get that fire started. And my dad always called that a catalyst. But that's a different kind of catalyst because that catalyst actually gets burned up or used up. But an enzyme is a catalyst that does not get used up. So it can catalyze a reaction and then catalyze another one and another one. It's not affected in the process. Enzymes are very specific, usually to a particular substrate. That's the molecule that they bind. So an enzyme and substrate fit together like you think of as a lock and key. So I have a key for my car. I have a key for my house, and they're not interchangeable, right? They go together specifically, just like there's a specific enzyme for a particular substrate. Now, the area, and we'll look at this a little bit in more detail in a second, but the area that the enzyme interacts with the substrate it's binding is called the active site. We'll look at activation energy and how enzymes lower that. That's the whole purpose. That's how they catalyze a reaction, or they cause a reaction to happen, because they're lowering the activation energy. One thing to keep in mind as you go through the semester and you learn things in this class, enzymes typically are named with the end ASE. So one enzyme we'll talk about today that has to do with your virtual lab is Catalase. So ASE ends in ASE. Another one that you may be familiar with is lactase, which is the enzyme that some people don't continue to make as they become adults, and therefore they can't break down the sugar in milk called lactose. Now, enzymes are going to all have optimum pH, temperatures, and salt concentrations that they operate or function at ideally, or the best. Outside of that range, it's going to affect their ability to carry out the reaction or catalyze the reaction. And at some point, if it is too high or low of a pH or temperature or salt concentration, it may affect the folding, because remember, the enzyme is a protein. Protein's three-dimensional shape is very important to their function. So if a temperature or pH or salt concentration is too far outside of the optimal range, it may cause the protein to fold incorrectly or become misshapen. And in that case, we call that denature, and it no longer can carry out its function. Enzymes have helpers called cofactors and coenzymes. We'll talk about those in a little bit. They also have inhibitors, which can prevent them from binding to their substrate. Feedback inhibition is something that happens very often in our cells, and we'll look at why that's important and how it uses negative feedback. Real quick, I just want to talk about chemical reactions. So this here is just a generic chemical reaction. And we have an arrow that usually represents which direction the chemical reaction is occurring. And on the left side of the arrow, in this case, the A and the B, these would be the reactants. So what we're starting with, right? And then over here to the right side of the arrow, this would be the product. Now in this reaction, I have two reactants and one product. It doesn't have to be that way. It could be A plus B, B, and we could have two products, C plus D. So I'm just wanting to make sure we understand reactants are on the left side of the arrow, products are on the right. So this figure is going to show us a little illustration of the structure of an enzyme and how it fits specifically to its substrate, the one that it binds. And so in gray here we have the enzyme. The substrate is shown in green. The area of the enzyme that makes contact with the substrate, that's going to be what we call the active site. So we see in this case the enzyme is binding to the substrate. When they're bound together, it's called the enzyme-substrate complex. And then the reaction is catalyzed. So the enzyme is helping to lower that activation energy so that the reaction goes forward. And we see in this case, we started out with a larger molecule as the substrate, and the reaction that was catalyzed produced smaller products. So is this going to be anabolism or an anabolic reaction, or catabolism or a catabolic reaction? Yeah, it's catabolic, right? Because we know that catabolic reactions go from larger molecules, break them down into smaller ones. Now, I already mentioned that the way an enzyme functions is it lowers the what we call energy of activation or activation energy in a cell. And what that means is, we talked in a previous video that some reactions are going to be endergonic, meaning they require an input of energy. Some reactions are going to be exergonic, which means they will release energy. No matter whether it's endergonic or exergonic, there's a certain amount of activation energy that must occur in order for the reaction to go forward. So what we see is in purple on this diagram, this high peak, that's the amount of activation energy that's required without an enzyme. The enzyme, however, lowers that to where the red dashed line is. So in other words, the enzyme, by catalyzing the reaction, it makes it more likely for this reaction to occur in the cell. Now, I mentioned to you that enzymes have helpers. So they can be in the form of cofactors or coenzymes. And the biggest difference is cofactors are inorganic. Remember what an organic molecule is? That's something that contains carbon and hydrogen. So inorganic molecules that help are things that you may take supplements for, or you may just get them in your diet. So copper, magnesium, calcium, those minerals are all examples of cofactors. Coenzymes are organic, and these are also things you may think about in supplements, or you just may think about in the kind of food that you eat, that these are important. And the reason they're important is because they're coenzymes for many of your important enzymes in your body. So, for example, vitamin B1. We will talk a little bit as we go through. We've already touched on ATP and how it's our energy currency. So B1 is important in us generating ATP from some of our nutrients. Vitamin C, we probably know some of that already as an antioxidant. It helps with our collagen production. That's just one function of vitamin C. Folic acid. So this is a very important component for pregnant women because it helps to reduce the incident of spina bifida and other spinal developments that may go awry. So it also is important in red blood cell formation. Vitamin A, we know, right? Maybe as a child, eat your carrots, so it's good for your eyes. We know that that's important in our vision pigments. Now, there are inhibitors of enzymes, and they can come in a couple varieties. So competitive inhibitors are just like what they sound. These are competing with the substrate to bind to the enzyme. So if the inhibitor is bound to the enzyme, then there's no spot for the substrate to bind. A non-competitive inhibitor is one, it doesn't bind at the same site as the substrate, so it binds to the enzyme, but in another location. However, by binding, what it does is it causes enough of a change in the shape of the enzyme that it no longer can bind to its substrate. That's also called an allosteric inhibitor. I have a few examples there for you. So carbon monoxide, now that's not the same thing as carbon dioxide, right? Carbon dioxide is what we breathe out. Carbon monoxide, or CO, that is a dangerous gas that can be produced from a car engine or from different appliances like your heater. Carbon monoxide, when we breathe it in, competes with oxygen to bind our hemoglobin. So that's obviously dangerous if we can't bind oxygen as we need to. Cyanide is an example of a non-competitive inhibitor. It binds to an important protein called cytochrome C oxidase in the mitochondria, which is one of the organelles in our cells, and we'll learn more about that in a future lesson. But it's important in generating ATP in the process that we go through called cellular respiration. So these are examples of how inhibitors can definitely cause problems for our cells. Now, inhibitors can also be helpful. So for example, penicillin, which we all know is an antibiotic, the way that it works is an inhibitor for a particular bacterial enzyme. So it prevents bacteria from being able to build new cell walls as they want to replicate. Now, this is an example of feedback inhibition. So first of all, I want you to understand that in our cells, we have many metabolic processes. And all I mean by metabolic process is it's something that is multi-step and it has multiple enzymes, different enzymes. So these are just generic, right? We have here no names, but enzyme 1, enzyme 2, and enzyme 3. So in this process, right, we have something that we're starting from. So this would be the initial substrate that they're representing with a green triangle. It's the substrate for enzyme 1. And then the reaction that enzyme 1 is catalyzing is the conversion of the green triangle to the purple rectangle. Then there's a second enzyme, enzyme 2, and it catalyzes the reaction from the purple rectangle to this blue odd shape, okay? And then enzyme 3 is a third enzyme in this pathway, and it catalyzes the reaction of the blue strange shape to the yellow square. Now, the important thing is, first of all, it's a metabolic pathway, so we have multiple enzymes, right, that are carrying out this. That's very common in our cells. The other thing I want you to see is that the end product, which is this yellow square, is actually serving, that should be an F, as a feedback inhibitor. So it has negative feedback to this first enzyme, okay? So this enzyme right here, when there is enough of the end product available in the cell, it is going to bind to that first enzyme. And notice when it binds on a separate site. It doesn't bind in the active site. However, by binding, it changes the shape of the active site so that the substrate can no longer bind. So that would be a non-competitive inhibitor or an allosteric inhibitor. And here's just another example for you to see how this can work in a cell. So we have an inhibitor that binds elsewhere. That's going to change the shape of the enzyme, and it no longer can bind the substrate. Therefore, the active site is altered. Now, on the right, what I want you to see is something we haven't discussed yet, and that is an activator. So in this case, it's necessary for it to bind. It changes the shape of the active site so that it can actually now, the enzyme can bind to the substrate. So the difference is, on the left side, this is an inhibitor that's changing the shape of the enzyme where it no longer can bind the substrate. On the right, we have an activator, which could be a cofactor or coenzyme that's changing the shape of the enzyme so that it can, in fact, bind the substrate. I mentioned to you that enzymes end in ASE. So here's one, lactase, or beta-galactosidase. This is the enzyme that you produce in your digestive tract, or some of you do, some of you don't. And the reaction that it catalyzes is it breaks down the lactose sugar, which is a disaccharide. Remember, di means two. So I have the structure for you here. Not important that you learn the structure of that specifically. I just want you to see that lactose is a disaccharide. It's found in milk. So if you drink milk or eat dairy products, this is a nutrient that's in that goes through your digestive system. The enzyme lactase converts lactose into one molecule of glucose, which is a monosaccharide, right? And one molecule of galactose, which is also a monosaccharide. Now, if you do not have this enzyme, you do not produce it anymore, then you're lactose intolerant, meaning you're unable to break down that disaccharide into the two monosaccharides. And it can cause, stomach issues and feel kind of uncomfortable. Now, they have products we can take, right, to help us if we have that issue. But this is an example of an enzyme that's occurring without you even realizing. Okay, the next enzyme we're going to talk about is catalase. And notice, again, the name ends in ASE. This is the enzyme that your virtual lab activity is going to examine. Now, this is a very common enzyme in many living things. And the reaction that catalase catalyzes is the conversion of this molecule here, H2O2, which is hydrogen peroxide, and it converts hydrogen peroxide into H2O, which is water, and O2, which is oxygen gas. So I'm just going to put in parentheses here that oxygen then is going to be in the form of a gas. Now, what I want you to focus on in this test tube on the left is we can see below this line right here, about 5 milliliters, there's liquid. Okay, so what was put into the test tube, what that liquid includes there is hydrogen peroxide plus the enzyme catalase. And what you see above that, it may be hard to see this white segment right here. This is just bubbles. This is gas bubbles. So this represents the amount of oxygen gas that was produced from the reaction. So in the virtual activity, what you're doing is you're examining whether or not the catalase enzyme is going to react with different substrates, or whether or not hydrogen peroxide is going to react with water to produce any reaction. And the way you're going to look for that is if there is a presence of bubbles, that would indicate the presence of oxygen gas.